Third talk in the RA session, a topic that used to be hot like 10 years ago, and then it kind of fell off the radar. But someone who's been keeping an eye on it and now is going to all of a sudden get increased importance this year, and that's biosimilars. Very pleased to have Professor K with us today. Jonathan, welcome.
Thank you very much, Artie and Jack, for this invitation. It's a pleasure to be here, and I've enjoyed the meeting so far. I'm going to talk about biosimilars back on the agenda. These are my relevant disclosures. The learning objectives are shown here, and I hope to achieve these by the end of this talk. So I'm going to start off by talking about biosimilars in the United States, and then talk about variability, drift, and evolution of biopharmaceuticals. Following that, I'll talk about how biosimilarity is demonstrated, and then talk about interchangeability, and finally, the key benefit of biosimilars, which are the economic aspects. Biosimilarity has been defined by the United States Congress in the Affordable Care Act in 2010 as the biologic product is highly similar to the reference product, notwithstanding minor differences in clinically inactive components, and there are no clinically meaningful differences between the biologic product and the reference product in terms of safety, purity, and potency of the product. The European Medicines Agency issued guidelines that are similar to these earlier in 2005, and reading those and those regulations from other countries, I've come up with my own definition of a biosimilar, which is that it's a legitimate, a legal copy of a biopharmaceutical that no longer is protected by patent, which has first undergone rigorous analytical and clinical assessment in comparison to its reference product. But that's not sufficient, because biosimilar candidates undergo that evaluation, and that's published, but they also have to have been approved by a regulatory agency in a highly regulated area such as the European Union or the United States, according to a specific pathway for biosimilar evaluation. So the first question for polling: which of the following biologics does not yet have a biosimilar approved by the United States FDA? Adalimumab, etanercept, infliximab, rituximab, or ustekinumab? So ustekinumab is gaining majority. It's losing, it's gaining. So about 75%, 3/4 of you said ustekinumab. So the correct answer is ustekinumab. There's actually a ustekinumab biosimilar candidate by Alvotech that has been submitted to the FDA for review and approval. The United States has now, since the approval of the first biosimilar, filgrastim SNDZ, in 2015, there are now 40 approved biosimilars, of which 29 are marketed. The biosimilars that are approved to treat rheumatologic conditions are shown here. There are 8 adalimumab biosimilars, of which one is currently marketed. That's adalimumab ATTO, which was the first to be approved. There are 2 etanercept biosimilars, but neither of these have been marketed, and neither of these will be marketed until 2028, because Amgen was able to get an extension of its patent exclusivity in 2011 because they found a patent that hadn't yet been filed, and that gave them an additional 17 years of patent exclusivity. There are 4 approved infliximab biosimilars, of which 3 are on the market. Pfizer's biosimilar infliximab is not marketed because they acquired Hospira, which had its biosimilar, the Celltrion infliximab biosimilar, so Pfizer didn't want to compete with itself and shelved its infliximab biosimilar, licensing it to Sandoz for the rest of the world. And there are 3 rituximab biosimilars that are available in the United States. All biologics have normal batch-to-batch variability. At the time of its approval, the manufacturer and the regulators agree upon proven acceptable ranges of variation in each critical quality attribute that are established during development, and then the product is monitored throughout its lifespan to assure that variation in these quality attributes fall within these proven acceptable ranges of variation. As long as the attributes fall within these ranges, the product is felt to not pose a safety or efficacy risk to patients. There can be drift, and here are 2 examples of drift within the proven acceptable range of variation. The upper graph is a trend, a gradual shift upwards in that quality attribute, and the lower is a shift, a sudden change that remains stable. And these are due to unintended alterations in the manufacturing process, which may result in deviation of these product attributes. Evolution of a biopharmaceutical is due to deliberate process changes that are made by the manufacturer, such as implementing state-of-the-art technology or scaling up production, and these changes are known both by the manufacturer and the regulators. There, as of 2013, in this editorial by Christian Schneider from the EMA that was published in Annals of the Rheumatic Diseases, there were 37 such changes after the approval of Remicade, 21 after the approval of Enbrel, and 18 after the approval of Humira and others for other products. Such changes in the manufacturing process of biooriginators can result in differences compared to the originally approved product. These are comparison of pre- and post-change batches of rituximab, and on the left we see G0 glycan content, and as of 2010, there was a shift upwards with a threefold increase in G0 glycan, and this correlated with increased antibody-dependent cell-mediated cytotoxicity, function which, according to gastroenterologists, might have some role in the efficacy of the molecule for infliximab in inflammatory bowel disease. This is rituximab, however. The goal of biosimilar development is to develop a product that's highly similar to the reference product. So here we see the pre-change range, and here we see the post-change range. And the biosimilar manufacturer purchases commercially available lots of the reference product and reverse engineers it to come up with the biosimilar. So because the currently available lots of reference product are within the current reference product quality range, the range for control of the biosimilar product is this, and over the lifespan of the reference product, the quality range is at least the same, if not larger. So biosimilars are sort of like Hebrew national francs. They answer to a higher authority. They'll have a narrower proven acceptable range of variation if they're different from the reference product. So another question. Approval of a biosimilar by a regulatory agency requires A, comparable efficacy demonstrated in the disease for which the reference product is approved, B, equivalent efficacy demonstrated in all diseases for which the reference product is approved, C, equivalent pharmacokinetics demonstrated in the disease for which the reference product is approved, D, equivalent safety in comparison to its reference product, or E, demonstration of interchangeability with its reference product. This is a complex question, but I'll talk about the answers after you vote. People are changing their mind. Oh, the correct answer is only 13% equivalent pharmacokinetics demonstrated in the disease for which the reference product is approved. So the FDA guidance on demonstrating biosimilarity talks about a totality of the evidence approach to reduce residual uncertainty. You start off with a very robust assessment of structural and functional characterization of the biosimilar candidate in comparison to the reference product. They undergo over 100 different analytical and functional assays. This is then followed by a small non-clinical evaluation, possibly in animals for toxicity, but that is reduced as much as possible. And then the molecule goes into human pharmacokinetic studies, and if there's a biomarker available, such as for an insulin biosimilar glucose, pharmacodynamic studies. Clinical immunogenicity is assessed throughout, and then there's at least one clinical trial conducted in a disease which is most sensitive to detecting potential differences between a biosimilar candidate and the reference product, and then post-marketing information is gathered. So the FDA says that it intends to use a risk-based totality of the evidence approach to evaluate all available data. They look at all of these data together in totality and information submitted in support of the biosimilarity of the proposed product. They say that the need for additional studies at each step in this progressive approach will be determined by the degree of residual uncertainty that remains at each step regarding the similarity of the products. So, for example, for an insulin biosimilar, if the pharmacokinetic and pharmacodynamic studies establish biosimilarity, there might be no need for a clinical trial. And as there's more experience gathered with biosimilarity evaluation, the extent of the evaluation may be lessened. So the general principles of demonstrating biosimilarity are that the biosimilar has been shown to be highly similar to the reference product in extensive comparative analytical studies. So the biosimilar has to demonstrate similar efficacy and safety compared to the reference product, as I mentioned, in pharmacokinetic, pharmacodynamic, and immunogenicity studies, and in at least one clinical trial. And no differences are expected in safety or efficacy between an approved biosimilar and its reference product. And because we have extensive clinical experience with the reference product in all of the indications for which it has been approved, and a biosimilar is now shown to be like another batch of the reference product, albeit made by a different manufacturer, clinical efficacy and safety have already been demonstrated by the reference product, so there's no need to demonstrate the efficacy of the biosimilar in all indications. One can extrapolate approval of the biosimilar candidate to all indications for which the reference product is approved without doing clinical trials in these additional indications. And that's where the financial savings in development of a biosimilar can occur. So the preclinical development of biosimilars involves cloning and production of the biosimilar candidate in a cell line to recapitulate all of the post-translational modification of the reference product, and then scaling up production. And then analytical studies compare the biosimilar candidate to the reference product in terms of structure, function, biological activity, and purity. The clinical development of biosimilars consists of clinical equivalent studies, pharmacokinetic and pharmacodynamic studies, and then at least one comparative effectiveness trial. These are data from the first published clinical trial of a biosimilar monoclonal antibody, the clinical equivalent study comparing the CTP13 biosimilar candidate for infliximab to reference infliximab in ankylosing spondylitis. This study enrolled 250 patients with active ankylosing spondylitis randomized 1-to-1 to receive either the biosimilar candidate or reference infliximab at 5 milligrams per kilogram infused intravenously over 2 hours per dose. The loading regimen was the standard loading regimen for infliximab, 0, 2, and 6 weeks, and then maintenance every 8 weeks up to week 30. The assessment was a pharmacokinetic assessment. The ratios of the geometric means of the primary pharmacokinetic parameters between weeks 22 and 30, and secondary endpoints included ASAS-20 and ASAS-40 at week 30, and safety, the incidence of adverse events. The primary endpoint was met. The ratio for the area under the curve tau was 1.05, and 90% confidence intervals fell within the equivalence range of 0.8 to 1.25, which is plus or minus 20% log transformed. The maximal concentration at steady state also met those criteria. And you can see here that at the 3 time points measured, weeks 14, 30, and 54, the ASAS-20 and the ASAS-50 tracked very closely together. However, these were on the plateau phase of the pharmacodynamic response, so these are time points at which you'd expect the 2 molecules to track together given the insensitivity of these clinical outcome measures to detect potential differences between drugs. In terms of safety, CTP13 and reference infliximab were comparable in terms of safety. A comparative effectiveness trial is shown here, an example of Samsung Bioepis' etanercept biosimilar. When it was a biosimilar candidate, it was known as SB4, compared to reference etanercept in rheumatoid arthritis. Here, the study was powered for an ACR20 at week 24 with an equivalence range of equivalence margin of plus or minus 15%. So the study enrolled 600 patients who were randomized 1-to-1, patients with active rheumatoid arthritis despite methotrexate, who received either the biosimilar candidate or the reference product at 50 milligrams weekly subcutaneously for 52 weeks. Primary endpoint, as I mentioned, was the ACR20 at week 24. Secondary endpoints are shown here, and also the incidence of adverse events and serious adverse events for safety. Here they measured time points, at early time points, as well as at the plateau phase. And you can see that the early time points, which are most sensitive to detecting potential differences between the biosimilar candidate and the reference product, track very closely together for the ACR20, ACR50, and ACR70, which you see looking from top to bottom. In terms of related treatment emergent adverse events, the etanercept biosimilar and reference etanercept are highly comparable, except for the injection site reactions, which were much lower, 3.7% for the biosimilar compared to 17.2% for the reference product. So a biosimilar has to be equivalent in efficacy and comparable in safety or immunogenicity, but it can be superior in terms of safety or immunogenicity and still be biosimilar as long as it is not superior or inferior in terms of efficacy. So interchangeability. The FDA the United States Congress put together this designation of interchangeability as part of the Affordable Care Act in 2010, but it wasn't until 2017 that the FDA came up with draft guidance for how to achieve this designation of interchangeability. And to do that, a sponsor must show that the proposed interchangeable product first is biosimilar to the reference product. So it first has to be approved as a biosimilar, and then that it can be expected to produce the same clinical result as the reference product in any given patient and in all of the reference product's licensed conditions of use. And for a biologic product that's administered more than once, the risk in terms of safety or loss of efficacy of alternating or switching between the biologic product and the reference product is not greater than the risk of using the reference product without a switch. So the study design that was proposed was a lead-in period of treatment with the reference product, followed by a randomized two-arm trial comparing a switching arm to a continued treatment arm, a non-switching arm, with at least 3 switches, with each switch crossing over to the alternative product, ending up on the biosimilar in the switching arm. So this is the schematic diagram. All subjects are on the reference product, and then they're randomized either to the non-switching arm or the switching arm, where they have switches, 3 switches back and forth. They have an adequate washout, and then pharmacokinetic endpoint sampling occurs, and they end up either on the biosimilar candidate, or the approved biosimilar, the interchangeable biosimilar candidate in the switching arm, or they remain on the reference product in the non-switching arm. The primary endpoints for such a study have to be pharmacokinetic endpoints, because those are much more sensitive to detecting potential differences than are the clinical endpoints. So area under the curve tau, and for subcutaneous molecules, the maximal concentration at steady state. And these are subjected to the standard pharmacokinetic equivalence range of 0.8 to 1.25. Secondary endpoint includes other pharmacokinetic parameters, safety, immunogenicity, and efficacy. And they suggested that U.S. licensed reference product has to be used as a comparator unless there's a bridging study between a non-U.S. comparator and the U.S. product. Post-marketing data is also included. So the first interchangeable biosimilar was approved in July of 2021, an insulin glargine biosimilar. And then Boehringer Ingelheim subjected its adalimumab biosimilar to a switching study compared to reference adalimumab in patients with chronic plaque psoriasis. And this has been published now in the American Journal of Clinical Dermatology, and it follows the FDA interchangeability study design. And what they found were that the area under the curve tau and the maximal concentration between weeks 30 and 32 fell within the predefined equivalence range of 0.8 to 1.25. Also, efficacy was shown to be highly similar, with PASI scores for both arms being highly similar. The immunogenicity was highly similar, and safety, again, was comparable. So this molecule was approved as the first interchangeable biosimilar monoclonal antibody in October of 2021. But it's not yet available because there's only one adalimumab biosimilar that's been marketed so far. Ranibizumab EQRN is the first interchangeable biosimilar that was approved by the FDA without switching studies. This is a vascular endothelial growth factor inhibitor, and because it's injected intraocularly, the manufacturer argued that the systemic pharmacokinetics would be irrelevant to what's going on in the eye, and hence it was approved both as a biosimilar and as an interchangeable biosimilar initially, without having to undergo a separate second stage approval process. Switching to the economic aspects to conclude this talk, the average sales price includes the discounts and rebates that are provided to PBMs, pharmacy benefit managers, and CMS posts these quarterly. And I've plotted for infliximab reference product in blue, infliximab DYYB in orange, and infliximab ABDA, the other biosimilar infliximab, in gray, the average sales prices. And what you can see here is that during the first 2 quarters of its release, the biosimilar infliximab, the first biosimilar infliximab, was priced about $200 higher per 100 milligram vial than the reference product, but that quickly changed. And then in the first quarter of 2018, when the second biosimilar infliximab was released, you can see that there was competition and all of the infliximab prices dropped at a more rapid rate. Interestingly, during the last 3 quarters, the average sales price of the second biosimilar infliximab, the one in gray, has gone up higher than both the reference product and the other infliximab. This is due to loss of discounts and rebates. There are now 8 adalimumab biosimilars that have been approved by the FDA, and they're all going to be launched this year. AbbVie reached agreements with multiple developers which forestalled market entry in the United States till this year, whereas they'd been released in Europe and other countries as of October 2018. AbbVie granted a license to each of these companies, so AbbVie gets royalty for every sale of the biosimilar adalimumab. It's sort of like a Kevin O'Leary deal on Shark Tank. So in July, we're going to have 6 adalimumab biosimilars that are released, and then finally Sandoz's biosimilar adalimumab is going to be released in September. The adalimumab sales have been forecasted to slump next year, so these have reached over $20 billion a year as of 2022. In 2023, they're estimated to drop to about only $10 billion or $8 billion, and then in 2024, they're going to drop even further. The growth in sales of the adalimumab biosimilars are expected to increase. In 2023, the first adalimumab biosimilar that's been released by Amgen is expected to generate about $450 million of sales, and these sales are expected to grow by 2026 to over $900 million a year, and the other adalimumab biosimilars shown here are expected to grow as well. The impact of brand losses of exclusivity in the United States as of next year are going to be about $14.2 billion loss of sales because of loss of exclusivity for all of the biosimilar, all of the drugs for which there are biosimilars. So in summary, all biologics are subject to variability, drift, and evolution, both reference products and biosimilars. A biosimilar is highly similar to its reference product without clinically meaningful differences. Rigorous analytical methods must demonstrate their degrees of similarity, including structural, physicochemical, and biological analysis, safety, purity, and potency. Clinical trials have to demonstrate equivalence of the biosimilar to its reference product, both in clinical and pharmacokinetic studies. The interchangeability designation generally requires additional studies with multiple switches between the biosimilar and the reference product, and the availability of biosimilars is expected to result in significantly lower spending on biooriginators. So what do I tell my patients? I tell them that FDA-approved biosimilars have been compared extensively to their reference products in many ways and have been shown to have equivalent efficacy and comparable safety. And FDA-approved biosimilars like another batch of its reference product, and use of a lower cost biosimilar benefits others with your condition by allowing more people to be treated with an effective medication. Savings realized by using biosimilars may be redirected to pay for novel therapies that address unmet needs, but there has to be something for the patient. The patient has to have some sort of a financial benefit, perhaps a lower or absent copayment, and the provider who's expending additional effort to explain biosimilars to the patient should also receive some sort of a benefit, such as a lesser need for prior authorization, only if the pharmacy benefit managers buy in. So with that, thank you very much for your attention.